Welcome, everyone. Uh, Firstly, on behalf of the speakers and the SEMA Safe Fair, I'd like to acknowledge the traditional owners of the land on which we're meeting today, and I pay my respects to elders past and present and to First Nations people here today. So good afternoon, everyone. Thank you for joining today's webinar entitled Air and Ageing. I'm Jeff Morgan, a Centre for Safe Air Chief Investigator based at the University of Sydney, and I'll be chairing today's webinar. Older Australians are disproportionately affected by air pollution, yet the biological mechanisms and long-term health implications remain under-recognised in policy and practice. This webinar features researchers whose work is reshaping our understanding of how air pollutants influence ageing, neurodegeneration, and chronic disease. Each of the two speakers today will present for around 20 minutes, followed by a Q&A session after all presentations have ended. So please drop your questions into the Q&A session, and you can do that as we go, and they'll just accumulate in that Q&A session. If you can't see the Q&A tab, if you just look on the bottom of the Zoom window, there's a More button. You might need to click that before you see the Q&A button. Our first speaker today is Professor Tony White from the Berghofer Medical Research Institute in Queensland. Professor White obtained a PhD in neuroscience from Murdoch University in 1996, then undertook a postdoctoral position at the University of Melbourne, investigated Alzheimer's and prion diseases. He established a research group at the Department of Pathology, University of Melbourne, investigating the role of biometals in neurodegeneration. Tony received an ARC Future Fellowship in 2011 and was recruited to QMI Berghofer in 2016 and was awarded an NHMRC Senior Research Fellowship in 2017. His research has contributed to the development of first-in-class metal drugs as a potential new therapeutic approach to treat brain disorders. He's currently developing new human patient brain models and applying these to understanding the impacts of air pollution and bushfire smoke in brain health and disease. He's also overseeing studies that aim to extend the cell-based findings of bushfire smoke health impact to firefighters in the field and the broader community. Over to you, Tony. Would you like to share your screen, please? Okay. Great. Looks perfect.
Fantastic. Thank you. So thank you very much for the invitation to speak here today. Again, I would also like to acknowledge the traditional custodians of the land and the elders past, present and emerging, particularly for the land that I live on and where I work on. Okay, so there's a growing body of evidence out there to show that air pollution has important impacts on human brain health. There's a lot of studies coming out, particularly over the last 10 years or so, to show that there's air pollution causes changes to memory function, or what we call cognitive decline, inflammation in the brain, impaired development of children's brains, they affect mental health disorders, stroke, and all sorts of other issues in the brain. So it's a very big area now, and there's a lot of evidence of that. There's also support that bushfire smoke also impacts these areas. To date, most of this work has come from studies on animals where they've exposed them to bushfire smoke and found a lot of changes occurring in the brains of these animals. But also some other studies that have linked exposure to wildfire smoke to changes in cognition and brain activity and so on. And particularly, there's a growing relationship between bushfire smoke and dementia. And dementia is now the leading cause of death in Australia for both men and women, ahead of heart disease and stroke and so on. So, really due to the aging population and also the advances that have occurred in those areas, that dementia is now the key killer in Australia. And it has also been linked to exposure to air pollution. One of the interesting studies that came out in the last couple of years is that air pollution was second on the list of factors that can lead to changes in the brain that are likely to be involved in dementia or Alzheimer's disease. These are modifiable factors, so things that you could potentially change. Diabetes is the most important factor, and then pollution, and then alcohol. So it's a really important factor in terms of how it affects our brains and our risk for dementia. So we know that bushfire smoke, or wildfire smoke, that's it elsewhere, as they call it elsewhere in the world, is full of a lot of components. There's thousands of different chemicals in there. And as I said, a lot of this work has been done on looking at how the bushfire smoke affects animals, but there's still not a lot of understanding on how it affects humans. There's been some recent papers, and one of them sort of looks very complicated here, but it's really just a paper that showed that the types of particles that occur in bushfire smoke, which are called particulate matter 2.5 or particulate matter 10, that's just related to the size of the particles. They are able to disrupt the blood-brain barrier. And this is a barrier that exists between our blood vessels and the brain, which protects the brain. And if you disrupt that, then all sorts of compounds can get into the brain and cause damage. And these people have shown recently that this can actually happen. It was something that we thought could happen, but now we know it can happen. And of course, the smaller the particles are, and there's a lot of what they call ultrafine particles, very, very, very small particles, like, a thousandth of a size of human hair, are able to get deep into the body and deep into the brain and have effects on our health. So what we want to understand is how wildfire or bushfire smoke affects human brain cells, and whether this leads to changes in brain function, and is it associated with diseases such as Alzheimer's and dementia, and therefore, finding ways that we could potentially intervene and stop this from happening. So there's a lot of compounds that I mentioned that occur in bushfire smoke, the whole list there on the left. They induce oxidative stress and inflammatory changes in human brain, and therefore can have subsequent effects on our health. So what we've been looking at in our laboratory is smoke that gets into the brain either through the olfactory system. So obviously our olfactory system is the nose. So you breathe smoke in through your nose, and it can actually directly go straight into the brain via pathways that occur from your nose to the brain. Or you can breathe it into your body, into your lungs, into the bloodstream, and then from the blood can go also into the brain. So there's different ways that smoke particles can get into the brain. So we've looked at neural cells. These are brain cells that we grow from stem cells. These are human brain cells, and also nasal cells. So as I said, the ones that are most likely to be initially exposed to bushfire smoke. We collect the bushfire smoke from burning material, put them onto these cells, and then collect the material and analyze it using a number of different methods. We've also set up a process where we can look at what we call microglia. So these cells are the only immune cell type that occurs in the brain. So there's a specific immune cell type in your blood and elsewhere in your body. There's lots of immune cell types, but in the brain, there's pretty much only one called the microglia. And so we've been looking at how they are affected. So to look at the bushfire smoke extracts, we, with our collaborators, Patrick Asari from Adelaide University, It's been burning vegetation material from Australian bush and collecting the smoke into what we call media. This is nutrients that we put on the cells, and comparing this to diesel engine pollution that we do the same with. And we see that when we treat the microglia, the brain immune cells, we get changes. The sort of purpley blue graph here is the diesel engine pollution causing an increase in nitric oxide, which is oxidative stress marker, much more so than the bushfire smoke, which is interesting because we actually see some opposite effects later on, which I'll talk about. And in neurons, human neurons, we see an increase in nitric oxide generation. So there's no doubt that the bushfire smoke can have strong impacts. It includes cell death at high concentrations in these cell types. We also see inflammatory markers. So again, this is inflammation that can occur in the nasal system or in the brain. And here we see these are what we call interleukins. These are very important inflammatory molecules that are released in response to toxins. And we see that occurring in nasal cells that are exposed to the smoke. One of the very interesting things we saw is that when we compared exposure of what we call BFSE, the bushfire smoke extract, compared to the diesel engine pollution on the brain immune cells from young people, people under 30, we didn't, after 4 days, we didn't actually see much of an effect with anything. So neither the diesel engine pollution or the bushfire smoke. But when we looked at the immune cells grown from older people, I think people over 50, we actually saw very strong effects with the bushfire smoke on inducing these inflammatory markers, IL-33, IL-8, and so on. So these interleukins, which are very important toxic markers in the brain, were very highly increased in our cultures exposed to bushfire smoke, but no real change with the diesel engine pollution. So there's something happening there with exposure of immune cells, brain immune cells from older people to the bushfire smoke, which is we think is really interesting in terms of understanding changes in things like Alzheimer's disease. For example, the IL-23, which is interleukin 23, which is a molecule that's very important in Alzheimer's disease, and that was one of the major increases compared to no change with the diesel engine particles, but only in older people. So we think that this is something that could be driving changes that could potentially lead to Alzheimer's. This very complicated schematic here is just really to show that bushfire smoke can induce oxidative stress, and you have a whole lot of markers changing that what we call heme oxygenase 1, which is an antioxidant molecule that's generated in cells. We think this is being upregulated to try and shut down this toxic pathway. So you've got a whole lot of toxic processes happening, and you've got cells responding by trying to stop that. So it really becomes a situation of where the cells become overstimulated with the toxic pathways, and they can't control them anymore. We also looked at some of the differences between different smoke types from pine, eucalypt, and some other Australian smoke on lung macrophages. So again, these are very early affected in the exposure to smoke. And what we show here is at the top here is just showing that these are changes, or not changes, but this is the baseline of what you see in cells, and this is exposed to the smoke, and you see the gene changes. So there's lots of gene expression from exposure to the bushfire smoke. But interestingly, we see quite different changes. So all these red markers here are the genes that are upregulated, whereas blue ones are downregulated. So we see these upregulated ones with in eucalypt, downregulated in pine here, and upregulated in mallee, and so on. So there's quite significant differences between different bushfire smoke types when they interact with our human cells. And a lot of these genes that are changed are related to oxidative stress, which is also seen across a lot of the different smoke types as well. So while there's differences, there's also important similarities that we could target. And these are just pathway studies that support what we've been saying. We've also looked at the effect of smoke on human brain organoids. These are mini brains, and we see high levels of toxicity induced from the bushfire smoke compared to diesel engine pollution in, again, the nasal cells. And one of the interesting things that we saw was when we looked at the most common genes changed in olfactory cells or nasal cells, that a lot of those cells that were, some of those genes that were changed in these olfactory cells were by the bushfire smoke is related to cognition or memory function, and but also related to air pollution. So there's certainly a relationship there between those two. Again, this is just looking at exposure to exposure of brain organoids or mini brains. And one of the things that we saw was a whole lot of changes to metal pathways. So we thought, well, maybe they're being exposed to metals in the bushfire smoke. We didn't really see a lot of changes to a lot of high levels of metals in the bushfire smoke. But it might be a combination of all the metals, or it might be changes to the cells that are causing changes to metals in the mini brains. This is something we're investigating. But we're trying to now drive this work into looking at how this affects people. And one of these areas are wildland or bushland firefighters. And we're doing studies to look at how the bushfire smoke affects them. So we look at them before the fire, do questionnaires, urine samples, and brain testing for their memory function. And then immediately after a fire, we do the same thing. And then after the season, we do the same thing again. We're currently measuring the oxidative stress and inflammatory markers in their urine samples, which we think that will give us an indication of just how much smoke they've been exposed to. The cognition studies are measuring things like short-term memory, ability to reason, concentration, and so on. So things that are also very important for firefighters when they're on the front line. We have done preliminary tests with this, and we are able to get data from it. It's quite variable at the moment, but the data is actually from a small number of people, and we're got 40 or more participants now. And so we were able to do this and get some very high quality data and compare this to the amount of smoke they've been exposed using particulate monitors, which these are graphs from particular people. And you can see that some of them are exposed as high as 4,000 or 6,000 micrograms per cubic meter, whereas the sort of recommended level is about 20 to 25. So for short periods, they can expose to very high concentrations. And so we're trying to map that back to what their roles are out in the field and how that is related to their cognitive function and their urine markers. And something that is being led by Michelle Lupton at QMR Berghofer, and she's driving something called the R-Fire Brain Health, and this is the Rural Fire Brain Health cohort that she's developing. And so what she's doing is getting a cohort of people together and collecting blood samples from them and from the firefighters, and looking at markers of oxidative stress and brain diseases and so on, and comparing the sort of clinical data that she has access to their medical histories, and comparing the relationship between the clinical data and biological markers to see whether there are ongoing changes to their health by being exposed to bushfire smoke. So just kind of taking what we're doing in the cells to see what's happening in people who are on the front line. And this is the appeal that raised the money for this, and it's been a very exciting project that's just getting up and running now. And it's a team being driven by Michelle, Jody, and Jessica, also with the help of Jeff Morgan and Jürgen Fripp and Duan Wilsons, and funding from various sources. Something else that Michelle is doing is expanding this, working with Jeff Morgan to try and understand the sort of geographic or Australian population level exposure to bushfire smoke and how this affects their brain health by, again, measuring their cognitive ability using online testing, and then sort of comparing that to their exposure to bushfire over a long period of time and seeing whether there's a relationship there. So just to summarize, what we're trying to do is to understand how wildfire or bushfire smoke can interact with human brain cells and nasal cells, which is the first cells exposed, and how this relates to human disease, and then trying to take this into the frontline populations of bushfire fighters, seeing what's happening to them, particularly in terms of their brain health, and more broadly across the community. And hopefully the work that we do can lead to changes in guidelines, particularly for frontline workers, but also for everyone to understand that it's more than just about the air quality. There might be air quality from different types of fires and different people, and we can sort of make those changes. And just like to thank a lot of people who've been involved in this work. That there's been sort of great teams getting together over the last few years to do this. And also particularly to Emily, who PhD student in my group, who's really been driving a lot of this work and is very enthusiastic about it, doing work out there in the field with firefighters and also growing cell cultures at the same time, and for putting a number of these slides together as well. So thank you.
Great. Thanks so much for that, Tony. That was fantastic. Now, don't forget to put your questions in the Q&A. At the moment, we've just got one question, so there's plenty of scope to get your questions answered. So don't forget to drop your questions in there, and we'll take them after our next presentation. And the next speaker is Zane Burnham. Zane's a final year PhD candidate at the Wicking Dementia Research and Education Centre at the University of Tasmania. He completed a Bachelor of Medical Research in 22 and now investigates how glucocorticoid stress hormones and bushfire smoke influence brain function and neurodegenerative disease. In 2024, he published his first paper demonstrating that glucocorticoid signaling can modify the intracellular effects of beta amyloid, producing both protective and harmful outcomes, and highlighting its complex role in Alzheimer's disease progression. Alongside his research, Zane is an active science communicator with young Tassie scientists, delivering engaging presentations to students across Tasmania during National Science Week. So over to you, Zane, and can you share your screen, please?
Great. Hello,
Great.
everyone.
That looks great. Fantastic. Thanks.
Great. So I'll be going through a little bit of my research, which explores dementia-related pathways, especially the impact of bushfire smoke and life stress. So, there we go. So what is dementia? So it's a very hard term that a lot of people think that dementia is just a disease that happens with normal aging, but this is quite misinformative. Dementia is actually an umbrella term which encompasses many different diseases such as frontotemporal dementia, Lewy body dementia, vascular dementia, and Alzheimer's disease. And what these all share are a decline in cognitive function that is severe enough to interfere with your daily life. And this is what distinguishes it away from normal aging. So in Australia, nearly 450,000 Australians are living with dementia in 2026, which is around 1 in 58 Australians. And in general terms, this is nearly the entire population of Tasmania, which is absolutely crazy to think that there's that many people in Australia living with the condition. And as Tony mentioned, in 2024, dementia was stated as the leading cause of death within Australia, and this includes Alzheimer's disease and the other dementia subtypes. By 2054, this number is expected to double. So between 1 in 40 and 1 in 48 Australians will be living with dementia. If we think of that as in a term of a suburban street, at least one person within that street may be living with dementia. So dementia is defined by various non-modifiable risks. So age, genetics, so 5% of dementia is familial, and also sex at birth. But there have been 14 potentially modifiable risk factors that are thought to contribute to 45% of dementia cases. So in early life, we see that as less education. In midlife, we see traumatic brain injury, physical inactivity, and smoking. And in later life, we go into social isolation, visual loss. And 3% of instances are thought to be caused by air pollution. So this has been studied, as Tony had mentioned. There's been lots of studies that have looked at air pollution and its effects on cognitive aging. But a lot look at not just a lot look at air pollution as in terms of traffic and diesel fumes and other air pollutants that may be within the environment. But let's just take a step away from that at the moment and let's look at something that's increasing across the world. So, bushfire smoke is one of the leading air pollutants that we are exposed to within Tasmania. With some of our clean air, it's great to be able to study bushfire smoke. And these are becoming more and more as part of the Tasmanian landscape. So we see that across recent years that fires have affected communities across Tasmania. And people might remember in the news back in 2013, the Dunalley wildfires, which were one of our biggest wildfires that we've had. And climate change is increasing the frequency and intensity of these wildfire and bushfire events. So modelling in 2014 showed that a lot of the east coast and northwest of Tasmania is more at risk of fire. So this FFDI is the Forest Fire Disaster Index. And it defines areas where droughts are affecting and where fire risk is increasing. And we can see that in recent times. So in Tasmania, nearly every week across summer, we were seeing different news articles, especially the Dolphin Sands wildfires, a wildfire in Launceston near Rokeby, different suburbs around Hobart. And this was nearly a weekly occurrence. But when bushfire happens, they can affect our lives and communities in various ways. So in smoke isn't just defined to a town, it isn't just defined to a border. Smoke from these Tasmanian bushfires on the northwest coast, you can see traveling across the Indian Ocean. And then wildfire smoke from Australian bushfires can be seen in New Zealand. The Canadian wildfires a couple of years ago, that smoke travelled into the northeast of the United States. And when this happens, we see, especially in Hobart, the sky becomes dark, it becomes brown, and you can visually see the amount of smoke pollution that's around the city. And similarly here. So within smoke, we have particulate matter. So Tony had already mentioned PM10 and PM2.5. So the PM2.5 are fine particles. They're around 30th the size of a human hair, so very, very small. These can be inhaled into the lungs, and because of their small size, they're able to enter the bloodstream and may have potential effects on the brain. So it's been thought that these PM components may be able to cross the blood-brain barrier through axonal transport of different nerves to reach the brain, and that also the volatile and semi-volatile organic compounds can activate sensory nerves within the respiratory tract that can lead to neurological disorders through the oxidative stress and neuroinflammation. So I want you to imagine for a moment that you sitting at your home having a coffee in the afternoon. You get a notification that a bushfire is has broken out a few kilometers away, and you decide to stay and defend your house from an approaching bushfire. You have 200,000 litres of water, you have a generator, you feel that you are ready to defend. You see that fire travelling over the hill and approaching you at 3 kilometres an hour with 80 km/h headwinds. This is what many of the community at the Dunalley bushfires back in 2013 experienced. And so you're not just being exposed to smoke. This photo went viral worldwide of families diving into the river to try and escape the fire. And not only are they being exposed to bushfire smoke, they're being exposed to a huge amount of stress. So stress hormone signaling within these people is severely increased. And a lot of these people also develop some post-traumatic stress disorder after the condition. So stress has been shown that it can contribute towards mild cognitive impairment and has been seen within many studies. So some within the World Trade Center, the earthquake and tsunamis in Japan. And we recently showed that stress hormone signaling can interact with amyloid beta in olfactory neurosphere-derived cells, having some influences on potential dementia pathology. So within that study that we'd done, we'd shown that around 23 proteins can interact with amyloid beta, with 16 proteins having a counteracting effect. So stress may be beneficial when amyloid beta pathology is present, whereas 7 have a more exacerbating effect. So stress on top of the amyloid beta pathology could increase deleterious outcomes. So what I wanted to do was investigate not only bushfire smoke and cortisol, our main human stress hormone alone on molecular mechanisms, but I wanted to look at their combination. So when I showed those modifiable stress factors earlier, they don't occur in isolation. They can have additive effects. They can, if you have social isolation, you may also have different risk factors that you are exposed to. So by looking at this combination, we're able to see whether there's certain modulatory effects of stress hormones on bushfire smoke. So I use an olfactory neurosphere-derived cell model. So this diagram is not too complex, but essentially it's taking a nasal biopsy and collecting olfactory ectomesenchymal stem cells. This isn't a completely clean culture, but they're cultured back into neurosphere-derived cells, which are the most accessible primary cell type that we have that can be gained from humans. And these were provided by the late Emeritus Professor Alan McKay-Sim. And I used 2 species of bushfire smoke. So I used eucalyptus and wet sclerophyll litter and Pinus radiata. So these are, if you've gone past any plantations, this is most likely Pinus radiata. It's a quick growing pine tree. And these 2 species of trees are primarily dominant across Tasmania. So something that I don't get to do very often as a lab-based scientist. I actually got to go and help burn materials, so it was a little bit of fun. You can see on the left-hand side that we have a tube collecting the smoke, and this is being bubbled through water. So we're collecting all the different particulates in water, and there's a vacuum assisting in this process. And so I then take this and freeze dry it down, and we result in that right-hand side photo of this very powdery substance. Very gross when it is suspended in water. Looks almost a urine yellow, but this contains particulates that are 2.5 microns and smaller, as we'd filtered during the process. And what my paradigm was that I grew cells within culture, and when they became 50% confluent, which means that the base of the cell culture dish was taken over by cells around 50% of the area, I added cortisol and different smoke solutions, so either pine smoke or a eucalyptus smoke. And after 48 hours, I collected the proteins from these cells, so the building blocks. And I use these proteins to do mass spectrometry. So again, it's a very complicated process that I've tried to put into enough of a timeline that you could understand. So protein extracts are broken down into peptides, so very small fragments. And these are inserted into a mass spectrometer, which can spray these proteins out to collect their different mass and also their charge. So by doing this, we're able to see different protein profiles, such as on the right, which can allow us to see what proteins are within a sample. And what I saw was for pine smoke versus control, there were various proteins that were changed. So IREB2, PPAT, and this is a volcano plot. So it allows us to see how much of a fold change. So we had a fold of 0.1 set, as well as p-value, so anything above a 0.05. But if you look at this graph and look compared to eucalyptus, it's a completely different set of proteins that are changed. So each species of wood has different compounds within it. Eucalyptus has the oils, whereas pine has different resins. But also these two trees burn at different temperatures. So different temperature burns can produce various different compounds as well. Looking between the pine and eucalyptus smoke, there's only 4 proteins that are changed, that are shared between the two tree species. So ATL2, XO44, MAP7D3, and TOP2B. But when we control for multiple corrections, this goes to a measly 1 protein from eucalyptus and 63 proteins that are changed by pine smoke. So this is again showing that vast difference that's between the two different species of trees. So for the rest of this talk, I'll mainly be talking about pine smoke, just because there's a lot more data that I can drive down with this. So I want to look at how pine smoke was changed when cortisol was absent versus when pine smoke was changed with cortisol present. And we see that there's pine smoke altered more proteins with the presence of cortisol, so that 65 proteins changed, whereas there were only 27 proteins changed by pine smoke alone, with 36 proteins being changed by both conditions. So we see on the bottom left-hand side that for proteins such as NQO1, pine smoke and with and without cortisol can increase the protein compared to control, whereas in ALK BH5 we see pine smoke can decrease. And collecting those full 128 proteins that are changed with and without cortisol, we see that pine smoke increases the abundance of 49 proteins, and decreases the abundance of 79 proteins. So this is just a heat map that explores how protein abundance can change across treatment conditions. And we see how it's very nicely clustered, which was great to see that there is that significant difference when pine is present and when it's not. So looking at these proteins, we found through functional enrichment that 8 proteins are involved in DNA replication initiation, which is quite severe for neurodegenerative conditions. So you don't want your DNA replication to not be working correctly. So diving further down into this, that we could explore whether there's problems with how different cells within the brain, such as microglia and astrocytes, may not be able to function correctly and may not be able to replicate, to, especially in dementia, where there is a loss of cells. So taking that back into some of the results that I'd gotten from the previous study, so looking at amyloid beta 40, which was our main changer, I wanted to look at those 128 proteins altered by smoke and amyloid beta 40. And while there is quite a minimal overlap, we do see that 9 proteins are induced by both amyloid beta 40 and pine smoke, which could present possible areas for therapeutic intervention in dementia, especially when someone's being exposed to pine smoke. And these proteins were caspase 4 and NF beta 2. So pine smoke, we saw that pine smoke may be able to exacerbate the effects in caspase 4, which could have more deleterious outcomes, whereas it may counteract the effects that we saw in NF beta 2. So it could be the fact that pine smoke does have some beneficial health effects, but there are so many more deleterious health effects that it also has on top of everything else as well. So want to wrap this up. So stressful life experiences and bushfire smoke, we believe, are potentially modifiable risk factors of dementia. They go hand in hand when you're in a area that's at risk of bushfires. And by understanding the pathways and mechanisms changed in dementia, we can inform policy and provide targets for therapeutic intervention. So this may mean in a bushfire event, do we move at-risk communities to evacuation centres further away? Do we make sure that at-risk communities have P95 masks, have HEPA filters within their house to prevent bushfire smoke from entering? Now, this work is not done just on the basis of one group. I'd like to thank my supervisors, Dr. Duncan Sinclair, Dr. Rachel Atkinson, and Professor Anna King, Dr. Richard Wilson at the Central Science Laboratory for all the help with spectrometry, as well as Fire Lab 3, Professor Faye Johnston, Megan Porter, and Scott Foister for all their help in helping me burn stuff, as well as Professor Tony White's group at the Cellular and Neurodegeneration Laboratory at QIMR in the Stem Cell group. Thank you.
Thank you, Zane. That was really fantastic. Great to have your work showcased at the Centre for Safe Air. We have a few questions in the Q&A, and so we'll launch into those. The first one I'll just throw to Zane. It's from Duan. Do you expect cortisol to have an interaction with other cell types too, and especially respiratory?
Yeah, definitely. So it's something that I'm looking to explore with further. So the cortisol response is a lot to do with the HPA axis, so hypothalamic pituitary adrenal axis, and it's primarily in the brain, but not just neurons, it could have an impact on different cell types, so microglia. And it may also have the impacts on respiratory. So not just through cortisol acting on it, but also that when we're stressed, our breathing rate increases as well. Um, so it could definitely have an effect there.
Great. Thank you. The next question we'll throw to Tony um, by Chris Cowie. Do you think there's such why do you think there's such a large difference in effect on cell health biomarkers between bushfire smoke and traffic pollution? Are the doses applied consistent with human exposures? Very acute for bushfire, shorter periods, chronic lower doses for traffic pollution, or more related to PM constituents, or both?
Yeah, I think it's a really good question. The dose that's been put on the cells is it's I think it's relatively similar. If you compare what people are exposed to in terms of traffic pollution, and I think the bushfire smoke extract that we put on them, we initially started off working from what we were told from a group that had worked with cigarette smoke, where it kind of was equivalent to something like smoking a packet or two a day or something. So, it's relatively high, but not ridiculous in terms of how much are they exposed to. It's not like standing out there in really smoky conditions for a long period. So I think it's probably more related to something in it, given how different the effects were, and also the differences that we see between young people and older people. It's probably not just a general smoke. It's probably something in there. I'm not sure what it is, and we'll have to try and narrow that down at some stage to see what it is. That then maybe we can target something in particular rather than just smoke in general. But yeah, it's a very good question.
Thanks, Tony. We've got a couple of questions now from Michael Abramson. Um, Bill, is it possible to bring in Michael's mic? He's got a couple of questions, or shall we go off the chat?
I think we can do that.
We've got a few minutes left, and Michael had a couple of questions. And being a humble epidemiologist, I'm very happy to flip some of these more detailed questions to Michael.
Okay, go ahead, Michael. Have we got you now? If we unmute you.
Hi. They were both great talks, and I'd like to congratulate both speakers for making their work so accessible to non-neuroscientists. My first question was for Tony. Sadly, some of these rural firefighters will die on the job, and their deaths will then be subject to a coronial inquest, often following a post-mortem examination. And I just wondered whether any consideration had been given to obtaining consent to examine their brains.
Yeah, that's interesting. It's something we hadn't thought about. I mean, there are a number of brain banks around the country that do collect brains from people who donate them. There isn't usually any kind of focus to try and get people in particular areas, usually more so in sort of disease for Alzheimer's or Parkinson's or something. But it's something that we could look at. And I don't know how many people you would sort of get, given that some people don't like to do that, and that's fine. So it would kind of be whether you can get enough to get anything statistical out of it. But I think it's a very interesting question and something that we should definitely think about. And we always need more brain donors across a lot of diseases and conditions and so on. It's something that we're always trying to do. So yeah, it's really interesting.
Thanks, Tony. Michael, while you're there, one more question.
Thanks, Jeff. My other question could be directed to either speaker, and that is, do we understand the chemical differences between, say, pine and eucalypt smoke? And I ask this because I wonder whether it might be possible to either plant species of trees or modify the existing plantations so that the smoke is less neurotoxic.
Zane, do you want to answer that?
Yeah, so I think there's some studies that have looked at it, but it's very difficult to find the complete composition between the two species. So, um, speaking to my friend in chemistry, because we thought, oh, it should be easy process to look into it. It's something that entire PhDs are being structured on to develop techniques to look at it. There's definitely going to be changes because eucalyptus trees burn hotter than pine trees. So these are able to burn a lot more of the compounds that are present within the wood, which is why I may be seeing those vast changes between the pine smoke and eucalyptus that I saw. So, um, yeah, I think there are definitely changes. I don't necessarily know what we could do further at this time, unless Tony has any other comment on that.
No, yeah, I don't really have much to add to that. It's again, it's an interesting concept, but trying to find what those compounds are and modifying tree species. But of course, a lot of what we're exposed to is coming from the vast forests that we have. So there's not really, smoke can travel a very long way, even around the entire globe. So it's very difficult to control where that smoke's coming from and what you're going to be exposed to.
Thanks very much. I think us epidemiologists appreciate that not all PM 2.5 is the same, and that goes back to Christine's question.
Hmm.
Yes, thanks, Michael. Well, there's no more questions. I'll jump in with a few very general questions of my own. So either of you, please feel free to answer these. Um, the first is, it's so important to understand the biological mechanisms that influence health, and this is really important work to get at that. But some of our work is very much at the sort of control end of air pollution and control strategies, and a big focus is PM 2.5. So how how long are we away, or can you see different control mechanisms coming from the work you're doing compared to the stock standard general PM 2.5 control, reduce exposure, and health messaging associated with that, with PM 2.5?
Well, maybe I could start with that. I think it depends a lot on where you're being exposed to smoke. So, it's quite different for people who are right on the front line fighting the fires where it's burning right in front of them, that are probably also being exposed to a lot of the gases and so on, which people who are more distant are probably mostly being exposed to PM 2.5. But then as smoke travels further, it becomes aged and interacts with oxygen and other molecules, and it changes and so on. So I think there's two aspects. One is how far away from the source you are and how long the smoke has been aged. And the other is what your own susceptibility is, which is something that we have to understand as well. And that's something that some people might be more sensitive to, and other people might be less, and vice versa. So I think over time, if we can sort of become more nuanced in the way that we understand smoke dangers rather than it's just, well, it's smoke or it's pollution and that's it. So Of course, even now, there's a lot of ultrafines, but we don't really have any legislation around ultrafines because they're very hard to measure. So because we can't measure them very well, we tend not to worry about them, but they might actually be more important than anything else. So it's a case of understanding what the different components are doing and then how to measure them and then get the legislation in place.
Zane, anything to add?
Yeah, I agree with Tony on
Right.
that. Yeah, it is very dependent on where you are. And as we've shown that different species are different as well. So it may be that we have to have different policy specific for different areas that may have different trees around them. So in Tasmania, we have Northwest Coast, which are more a rainforest subtype of tree, whereas down south you'll have a lot more eucalyptus. Is there something that we need to have that on specific areas? Are there specific guidelines that we need to adapt or alter for at-risk populations?
Thanks, Zane. And just quickly, perhaps, Tony, a frontline response when you can't really get rid of a source of pollution and people are exposed is personal protective equipment. How's your work playing into that with firefighters?
Well, that's the people that we've been collaborating with, Duane Bilsons, Kiam, Padamsi from West Australia and so on. So these people who are working on the sort of front line with the firefighters, trying to understand, what they need, because a lot of the time there may be sort of directed as to what you should be doing, but it's not always practical. So it's about getting out there with the firefighters and understanding what their situation is and how to deal with that. And it's, it is a difficult area, but something that we're all trying to do is to bring all of these areas together from, cell studies through to epidemiological studies and safety studies and so on, and everyone sort of talking together rather than one group or the other saying this is what should happen and the other group saying, well, no, it can't, and so on. So it's complicated, but at least I think the conversations are happening now.
Fantastic. Tony, thanks very much for that. Yeah, we've still got a way to go, I think. So just thank you both, Tony and Zane. My sincere appreciation and the appreciation of the Centre for Safe Air for sharing your time and insights on the topic. We hope our attendees found the discussion valuable and informative. We managed to get to all of the questions, but if you have any further questions or would like any additional resources, please don't hesitate to reach out and contact the Centre for Safe Air. And we look forward to seeing you all at our next webinar. Thank you and have a great rest of the day. Thanks, everyone.
Thanks.
Thanks so much.
Bye.
Bye.